I'm Eddy the Boer. I'm working in Department of Genetics of the University Hospital Groningen, and I work in the Innovation and Development team. And I will give this presentation with my colleague Arjan Buijs, who will introduce himself. We want to tell you today about our experiences with optical genome mapping in our department and how we resolve findings in leukemia with this technique. We want to start with the classical approach in our department. So before we introduced optical genome mapping, then we want to tell about the implementation study, how we implemented optical genome mapping. Then I will talk shortly about the guided assembly, the FIRE software. And then we will go to our present leukemia workflow and tell shortly about the pipelines. Then Arjan will tell you about some interesting cases from practice, and we will finish with the concluding remarks. Well, before we introduced optical genome mapping, we used techniques beside next generation sequencing for the germlines, and that are PCR techniques and Sanger sequencing. But in this presentation, of course, we will talk about the techniques that we use for hematological malignancies, and that are mainly array, FISH, and karyotyping. And these three techniques we try to replace with optical genome mapping. And we want to do this because using all these techniques is time consuming, expensive, and sometimes also difficult to maintain, and especially in knowledge, because of course we need to have technicians that can work with all these techniques. And I need to say these techniques also come with limitations, and I will come back to that. Then optical genome mapping. It is a technique where you can produce and analyze extremely long DNA fragments with high coverage. And this high coverage is necessary, of course, because we have somatic aberrations that can be present in a low percentage. And they can also be really complex, and for that reason it's important that we have long fragments. And the fragments you can see in a distribution have a N50 of about 400 kb, and as I told already, they are there in a really high coverage. And we think optical genome mapping, and we also want to show that, of course, that optical genome mapping can combine several current methods in one technique. We started with an implementation study of 18 bone marrow aspirates, and in this implementation study, we did not use one pipeline because that FIRE pipeline was not there yet, but we used the de novo assembly and the rare variant analysis pipeline. With the de novo assembly pipeline, it was possible to have a higher sensitivity for small aberrations, as for example for small deletions. And with the rare variant pipeline, the sensitivity for the low present subclones is higher. We detected 23 OGM calls in this 18 bone marrow aspirates. 13 were confirmed with the classical methods array, karyotyping, and FISH, but 10 were missed with this current methods. Because of limitations in resolution of these classical methods. 5 of these 10 aberrations could also not be confirmed, detected exactly at the position where they happened. And this, of course, has to do with the limitations of optical genome mapping, because it is a method, a labeling method. So if there is a small deletion in between the labels, then it cannot be exactly localized. That is also not necessary for hematological malignancies in most of times, but I will show you that in the situations where optical genome mapping can exactly detect the aberrations, it detects exactly the aberrations, and otherwise, if it detects an aberration in between two labels, that the aberration is really there. So, and here is an example of a deletion in between two labels, and here you cannot say where the deletion is exactly located in between the two labels, while TP53 is part of this region, but not the whole region. For this, we did long-read sequencing, and we saw that there is indeed a 1.5 kb deletion in between these two labels. So optical genome mapping is correct here. And we could also see that the deletion is located before the TP53 gene, and that it is indeed, and that's really nice, a 1.5 kb deletion. So optical genome mapping is right in the size of the deletion, but can of course not detect the location because it's in between these two labels. Another situation was a translocation between chromosome 14 and 17 with an uncertain breakpoint. And with long-read sequencing, we saw that there was indeed a breakpoint in this purple location, in this uncertain location of optical genome mapping. And we could also exactly localize this breakpoint. And the breakpoint is before the gene instead of in the gene. Then, at a certain moment when it was possible to combine the de novo assembly pipeline and a rare variant pipeline in one pipeline, we decided to do this. Now we use the guided assembly, and we do this with an effective coverage of 300x. And with this pipeline, we can in fact detect the same as in one pipeline with both separated pipelines that we used previously. So the variant allele frequency is comparable with the rare variant pipeline, it's 5%. The resolution for insertions and repeats is almost the same with this new pipeline. And only the resolution for deletions is a bit lower. So it was 500 base pairs with the de novo assembly, but it's now 7 kb. But that is absolutely not a problem, and it is possible the same as with the de novo pipeline to see the loss of heterozygosity. And this is all concluded in really nice via software. Then we also try to analyze CLLs and MDS samples that we did previously with SNP array, and we tried to resolve this with the guided assembly. We detected all aberrations that we expected, so all 30 aberrations of all aberrations in these 30 samples, but we also detected 3 additional translocations and also 4 other additional aberrations, and we confirmed the additional findings. So here we show that the guided assembly detects all variants that we detected previously, but also 7 new aberrations. We have now 3 instruments in our department. So we have 2 Sapphires and 1 Stratus, and we use our optical genome mapping application for myeloid and lymphoid studies, and we detect all kinds of hematological malignancies with this technique. And we showed and also use it mainly as a all-in-one technique, and that was also a goal before we started the implementation study. We processed about 900 samples until now, and weekly we process about 26 samples, and that is 4 runs a week. And now Arjan will show you some interesting cases from practice.
Hello, my name is Arjan Buijs. I'm a laboratory specialist mainly in leukemia for many years. And I would like to show you some of the interesting cases we have. So I'd like to start with 3 cases of CML, chronic myeloid leukemia, that's characterized by 922 translocation, and that you see in the right upper corner in chromosome 9 contains a part of chromosome 22 and a small chromosome 22 derivative chromosome 22, the Philadelphia chromosome. The translocation results in juxtaposure of 3' able sequences, including the tyrosine kinase coding exons, to BCR on the Philadelphia chromosome. So the first example I'd like to show is here you see a case with a 922 translocation, and you see in the left hand corner the circle plot, the line between chromosome 9 and chromosome 22. In the upper right left corner you see the whole genome view from chromosome 1 to chromosome X, in this case a female. And on the right you see the able region on chromosome 9 and the BCR region on chromosome 22. And in this case you see that OGM demonstrates a balanced translocation 922 with a BCR able rearrangements also on the molecular level. It's a second example I'd like to show again, a circle plot demonstrating a 922 translocation. However, if we zoom into the breakpoint regions on chromosome 9, the able region, you see that we have loss of 5' able sequences. Well, for those of you who are doing FISH with specific probes, that's not uncommon that you lose your 5' signal. But also we see that the 3' BCR region on chromosome 22 on the lower panel is lost too. So what you can see with OGM, you see the translocation, but you can also run into loss of 1 or 2 of the non-oncogenic parts of the translocation partners, and that's not uncommon. The last sample I'd like to show to you is here. What you see, the software in the upper right corner calls a deletion of 3' able sequences on chromosome 9. Well, that's true that the software calls it, but it's not what we expect, because you need the 3' able sequences to get an oncogenic product. Again, you see in the circle plot there is a 922 translocation, but if we then zoom into the BCR region on chromosome 22, then you see that it is called as an insertion of 3' able sequences within BCR, resulting in a BCR able bona fide fusion, so a balanced insertion. Another case I think it's interesting to show is a case of MDS in which the chromosomes show an inversion on chromosome 3 in the long arm, as you see with the blue arrow in the right chromosome. And then if you look at what's going on there on molecular level, on the right part of the slide, then you have the GETA2 gene on 3q21, with its distal myeloid enhancer that is essential for GATA2 expression in myeloid cells. and then more distal you see the mecom locus. And because of the inversion, the distal myeloid enhancer of GETA2 inverts towards MECOM, thereby driving MECOM expression in myeloid cells, but losing GETA2 expression because of the loss of the myeloid enhancer. So this case, it has clearly an inversion by chromosome banding, but as you can see in the upper right corner, that there's what we use is also a fish triple column fish probe. And if you look at the left-hand corner, you see that there are two fusion signals present and no break part present of one of the loci. So it is what you expect. And here we go, zoom into the OGM. What OGM shows, and we zoom into chromosome 3, the Q arm. On the left you have the GATA2 locus, and on the right you have the MECOM locus. And what you see is that there's one big inversion, turning the GATA2 enhancer towards MECOM, but also on the right-hand side you have a smaller inversion with all the probes that we use for fish, and that smaller inversion turns MECOM towards the myeloid-specific enhancer. So indeed, we can say that OGM can elucidate more complex rearrangements, and in this case, it's a very high risk AML one with a MECOM rearrangement. And I would like to end with a case of myeloprotective neoplasm that has the classical JAK2 mutation, and that was progressing to myeloid fibrosis with an increasing allele frequency of a TP53 mutation in time. And this ultimately presented with a myeloid sarcoma, which is a extramedullary acute myeloid leukemia. So this myeloid sarcoma was analyzed with a next generation sequencing pan-cancer targeted panel, and that only revealed the already known JAK2 and TP53 mutations. So the question was, are there any other structural driving mutations present? So we did OGM on the DNA of this rare sample, and here you see the result. You see the whole genome view of the OGM. You see many tetrasomies and many trisomies and many structural aberrations. And what it revealed was a near triploid pattern without any known driver mutations or gene rearrangements. Well, near triploidy is a rare but recurrent pattern in AML, and it has a very poor prognosis, and this case we recently published. So, indeed, we have now for a year experience with OGM. We use two sapphires and one stratus. We are quite satisfied because we identified several rare recurrent translocations in various types of leukemia. And of course, those we think those who are cytogenetically not visible are the ones you want to see, and we see them with OGM. And we are very content with the latest versions of the VIA and the guided pipeline. So by this, I would like to mention the people who are involved, particularly Vincent Vroom and Anouk van der Veen, and many of the other laboratory specialists, but also the technicians who work for the arts. And if you have any questions, please email. Thank you very much for your attention.